Welcome friend and my dear student in the Sanjini Mahavidyalaya Sapulis online video classes. We are continuously organizing such nice online classes for students' career development. So let us start today's topic. Today we are going to study the part chemical stability of DNA. This part is very important because DNA is more chemical stable than that of the RNA. That's why we are going to see on what basis DNA is more chemical stable than that of the RNA. That part we will see. So DNA is a deoxyribose nucleotide. DNA is also called as polynucleotide because DNA is made up of a number of nucleotides. One nucleotide binds with another nucleotides. Likewise, there is a chain. That's why it is called as polynucleotides. DNA and RNA, basically they are made up of three components. One is the deoxyribose sugar or ribose sugar, that we can say pentose sugar. Second one is the nitrogen bases and phosphate group. The pentose sugar, in case of DNA, there is the presence of deoxyribose sugar, and in case of RNA there is the presence of ribose. In place of 2 carbon number of ribose there is hydroxyl groups, and in case of deoxyribose, in carbon number 2 there is the presence of hydrogens. So on the basis of the differences, is that's why it is called as deoxyribose. In DNA is called as deoxyribose sugar, and that's why RNA is not that much chemical stable due to the presence of hydroxyl group in carbon number 2. Apart from that, nitrogen bases, in case of DNA 4 nitrogen bases are present, and in case of RNA also 4 nitrogen bases are present. But in case of thymine, in the DNA thymine is present. Apart from thymine, uracil is present in case of RNA. So in the nitrogen bases, 2 nitrogen bases, they are pure in nature. Purine means 2 ring structures, and pyrimidine is a single ring structure. These are the 3 nitrogen bases. Adenine and guanine, these are double ring structures, and cytosine, thymine and uracil, they are the single ring structures. Adenine always pairs with thymine and guanine always pairs with cytosine in case of DNA. But in case of RNA, adenine always pairs with uracil and guanine always pairs with cytosine. And last one is the phosphate group. It is the main important component of the DNA. So nucleotide chains are antiparallel. It means that DNA is made up of 2 strands. One strand pairs with the another strands, and the direction of one strand, it is 5 prime to 3 prime, and another strand, it is a completely opposite directions, 5 prime to 3 prime likewise. And it means that 5 prime and 3 prime, it is nothing but it is the carbon numbers of deoxyribose sugar and ribose sugar. Sugar phosphate backbone are one on the outside of the double helix, and the bases are oriented towards the central axis. It is the sugar and phosphate backbones. It is a strong backbone, because due to which what happens, it gives stability in the DNA. Then adenine always pairs with thymine. There is the presence of hydrogen bond, double hydrogen bond. Guanine always pairs with cytosine. There is the presence of 3 hydrogen bonds. This is the combinations, and whatever the 2 strands of DNA, they are always equal in size. Distance between these 2 strands is equal from top to bottom due to that type of nature. You just see the 2 hydrogen bonds in between thymine and adenine, and 3 hydrogen bonds between cytosine and guanine. DNA doesn't have a reactive OH group like RNA, that's why they are stable. DNA is a double stranded, meaning main binding sites are already bound by another DNA. DNA is bound by histones, making it less accessible to reactants, and that's why DNA is more stable. DNA has repair mechanism. Repair means whatever the effect of any environmental factors on DNA, and if the DNA gets damaged, DNA itself has the ability to repair, and that's why, and after repair mechanism or repair process, DNA it becomes in original just like in the past. That's why this mechanism gives more important, due to which it is more chemical stable. RNA in the cytoplasm, which is full nucleases and other reactants. Apart from that, RNA is designed to be immediately used and then recycled. It means RNA is designed to be immediately used and then recycled. It means that in the environment, lots effect on RNA. That's why chemically it is not stable. That's why it is recycled. But DNA must last the lifetime of the cell and still be in good shape when passed to progeny. It means there is no side effect of on DNA, and it becomes same DNA in the shape as well as in the component without any change, and it transferred from parents to progeny without any change in the DNA. That's why it is more stable. Nucleotides are linked by phosphodiester bond to form polynucleotide. Phosphodiester bond, it is a strong bond, that's why it is more chemical stable. Covalent bonds between the phosphate group of one nucleotide and 3 carbon of the sugars of another nucleotide, it gives what? More stronger ability to DNA. This bond is very strong, and for this reason DNA is remarkably stable, and DNA can be boiled and even autoclave without degrading. It means without any change, the DNA it becomes same, it becomes without change. The ends of the DNA and RNA chains are not the same. One end of the RNA chain of the has a 5 carbon and another one is the 3 carbon atoms. You just see, this is the polar and negatively charged hydrophilic backbones of the alternating deoxyribose and phosphate group are on the outside of the double helix, and the non-polar hydrophobic bases with their planar structure are stacked inside the double helix. It means due to the hydrophilic and hydrophobic nature, due to the polar and non-polar nature, it gives more stronger, it becomes more stronger, because the charges, ionic charges, non-polar means it is equal ionic charges between the two nitrogen bases, and polar means there is the unequal charges, because due to the presence of two different kinds of the structures and whatever the nitrogen bases, whatever the pairing of nitrogen bases in the DNA, it present one above another just like a stack of coins, and they are equal because double ring structure always pairs with single ring structure. Likewise they are pairing, that's why they are the equal. The two strands of the DNA helix are held together by hydrogen bonds between the nitrogen bases. Just before I given the idea about how these pairs, nitrogen bases pairs with each other, and on the presence of what bonds they pairs. The double helix structures places the non-polar bases in the center surrounded by the charged phosphate, and due to the charged phosphate, what happens? the DNA has more stability than the RNA. So friends and my dear students, this is all about the chemical stability of DNA. DNA is more chemical stable than that of the RNA, and that's why we can use DNA artificially for any changes, and with the help of DNA we can produce hybrid varieties of any crop, plants, microorganisms, or any others. And DNA we can handle easily in the laboratory levels. uh, and what type of the changes we can change in the DNA, that we can carry out in the laboratory levels, but we cannot change in case of RNA. And with the help, that's why DNA is important. DNA, we can study more DNA as compared to RNA, and that's why it is more stable. So friends and my dear students, with the end of such a part, the chemical stability of DNA part is completed. So if you have any problems, you have to write any questions in the comment box of the YouTube channels. I am always ready to answering your questions. So friends, tomorrow we'll see the another part, that is the prokaryotic chromosomes. Till then, bye.